Thanks for the invitation to speak. Kind of a daunting process for me here to talk about all of the microbiome and human health and disease in about 25 minutes. So let's get started. So just out of curiosity, I mean, who's heard of the microbiome here? Everyone's heard of the microbiome. Who feels that their research involves, quote unquote, microbiome research in one way, shape, or another? So not as many people. So, I mean, it's often been associated with health, and there's some maybe unfortunate reasons it's been associated with health because NIH initiated the Human Microbiome Project. But I think it's very important now that we have the National Microbiome Initiative and that many funding agencies, USDA, FDA, CDC, DOD, beyond NIH, even NOAA, you know, I mean, we're talking about, you know, microbes on clouds and things like that. So that many people are very interested in this idea of the microbiome. But what are we talking about? We've passed the word around a little bit, and sometimes maybe it's the med school piece in me. We always have to define what our terms are. So when people refer to the microbiome, and it's already been referred to this way, if not necessarily explicitly stated, the idea of microbiome doesn't come actually from the word genome, but comes from the word biome when you build those little dioramas out of your shoe boxes, is that this is the total community and the environment that they inhabit. And I will try to use that term when I'm referring to the whole system. In many cases in the gut, that refers to the host, as well as all the microbes that are there. And if I'm trying to speak specifically about the microbes themselves, I refer to the microbiota, in which case those are the microbes. So what are these microbes? So we're focusing on the human body. It's interesting that you've chosen two physicians to speak at the end of this talk about food safety, because we are covered with microbes, many places. No place is really sterile. There's some suggestions that you can at least see if RNA is stable, as Martin said, DNA is particularly stable too. So we can find DNA in the brain, we find it in the placenta, we find it in places that we think is sterile. We're still kind of debating what to do with that. But when it comes to health and disease, there has been a lot of focus on disease, because every place that you find microbes, you see associations, you see changes in the structure of the microbiota related with a whole variety of different illnesses. And the problem is how many times is this an association and when is this causation? And how do we tease that out? So what I would like to do today is actually kind of present a case study that's more from the medical field, but can be applied, if you think about it, to anything related to food safety, microbes existing in complex environments such as a food matrix, such as inanimate objects, such as you might find in a processing plant. But we will start and spend most of our time on the gut microbiota, which is the largest collection of microbes in and on us. You always hear this term that, you know, 90% of the cells are microbes, or maybe it's 1-to-1. I think we're kind of debating angels on the head of a pin, but there are a lot of them. Let's put it that way. And they have increasing density in the gut, with the highest levels being found in the distal GI tract. But what can they be doing? It's nice that there are a lot of bacteria there, but what functions can they have? They can actually metabolize luminal contents. Gary is going to talk a lot about the diet-microbe interactions, but they also synthesize things such as vitamin K. What I'll be talking about is this idea of colonization resistance, how the indigenous microbiota of the gut can actually protect us from pathogenic organisms. And they're actually very important for education and immune signaling. So there's a lot of functions of the microbiota. And how can we study them? And I said, Gary's going to talk a lot about this, but don't just think of these as bacteria there. These are little factories, right? They have as much life and physiology as we do, and they can carry out a lot of very complex, and we got from Martin, some of the different kinds of conversions that they can carry out. And so this is how we have to think about it. When we think of the microbiota, we need to think about it like a forgotten organ, as Fergus Shanahan said. So this is a talk about next generation sequencing and why does the microbiome lend itself to next generation sequencing. But let's think back a little bit further. When we're studying a microbial community and their environment, what are we interested in? Well, one of the things we might be interested in is the structure of the community structure of the microbes. Or we may actually want to look at the metabolic potential, something that you can see through metagenomics. Or we actually, as Martin pointed out, we want to see what they're actually doing. What is their in situ activity? And there are a lot of different techniques that can be used here, including cultivation, which is not dead, and I'll bring that up in a little bit. But the advances in next generation sequencing have facilitated certain types of analyses where we can look at community structure, where we can look at community potential and also in situ community activity. And so I'm going to go over some of this that hasn't been covered already. And in particular, the idea of using the 16S, our RNA encoding gene, to look at the community structure of microbes. Martin already pointed out that this is a three-dimensional RNA, and because of that, it actually has very conserved regions that are conserved throughout most organisms that contain ribosomes, and therefore you can use broad-range PCR to amplify them. But there is variation in the stem and loop structures, so you can actually use those variant regions as sort of a fingerprint to identify bacteria. And how has this been used? I mean, most people have heard about this, but I'd like to take you in a little bit inside the box to let you know how this can be done. It's been pointed out that the sequencing is easy. It's the data analysis that can be hard. But it's not that hard if you understand some of the principles. So let's set the stage. I am an infectious disease physician. And for the past 150 years, we've been working on the germ theory, right? That pathogens, bacteria, viruses can cause disease. And Koch came up with these postulates as how to look at the causation of bacterial disease. You have to find a pathogen when you have the disease. You have to isolate it in culture. You have to give it to a willing medical student or a lab rat and recreate the disease and get the pathogen out again. And we've used this to prove causation of a lot of different diseases, smallpox, anthrax, tuberculosis. And because of that, as an infectious disease doc, and my hero, we come up with the idea that the only good microbe is a dead microbe. And we have a very powerful tool to kill microbes, and that's antibiotics. And we've used antibiotics quite a bit. We've also used them as growth promoters, not just for health, other things. But let me present a case that was first presented to me in medical school. We use antibiotics. You have a person who smokes a lot, has chronic obstructive pulmonary disease. He comes in, he might have an exacerbation of this, or he might have a pneumonia. So what the physicians will do is they'll treat him, quote unquote, broadly with broad spectrum antibiotics, a cephalosporin, a respiratory fluoroquinolone, and the patient gets better from the pulmonary standpoint. His breathing is better, but about 3 days into his course, he starts complaining about abdominal pain, diarrhea. In severe cases, he can go to the intensive care unit. He can actually lose his colon. What's going on? You were treating a lung infection with antibiotics. Why did the patient now all of a sudden develop a GI illness? Well, who's heard of Clostridium Difficile before, right? So we know that this is one of the side effects you can have from antibiotics. It's an anaerobe, it's gram-positive, it's a spore former. So we already heard about the difficulties in eliminating listeria from a food processing plant. If we ever had to try to eliminate C. difficile spores from hospitals, we would shut down hospitals because we cannot get rid of them. These environmentally stable forms, they're resistant to alcohol hand rubs. So if you use the alcohol hand rub, you dry off your hands so that you can transmit the spores to the next patient quite easily. You got to wash your hands, right? Now, it's present in only a small fraction of the normal population. So what happens? Why is that? Why don't we normally see C. difficile around? Well, sort of the cartoon that we like to talk about is that your normal microbiota actually mediates something that we like to call colonization resistance. And we'll talk a little bit about the mechanisms. But when you take antibiotics, and it could be for a skin infection, lung infection, you change the microbiota such that you're in a susceptible form. And if you get exposed to the C. difficile spores, they germinate. They'll become the vegetative form. This vegetative form produces a very potent toxin that can actually destroy the gut epithelium, and that's how you get disease. So you'll call in your infectious disease doc, and he'll say, well, you might have gotten in trouble with some antibiotics. We'll just give you more to get you out of it. We have antibiotics that target C. difficile, quote unquote, specifically, but they target a lot of other things as well, and most patients get better. But about 25% of patients, if you successfully treat them for C. difficile with an antibiotic such as metronidazole or vancomycin, when you stop all antibiotics, they'll recur. And you get these patients who go through this horrible recurrence disease cycle. 10, 20 times, they can lose, half of their body weight and die from this recurrent C. difficile that you can't clear. And we assume that's because the microbiota is so sufficiently altered that you never get back colonization resistance, because they can never get back a normal microbiota. So of course that prompted people to think about, how do we restore the microbiota? And you can see it down in the lower left-hand corner. How about fecal transplants? That should have a normal microbiota, and it actually works really well. It's been tried for a number of years, first reported in 1958, but it sort of changed a little bit in 2013 when the first trial using fecal transplantation for recurrent C. difficile showed an amazing success rate, 94% success rate, as opposed to 30% with just trying to give additional rounds of antibiotics. And so I'm going to use this setup to kind of demonstrate how we can look at the community structure using high-throughput sequencing with 16S. And so this is work that's done by a postdoc of mine, Anna Seekatz, where she wanted to see, okay, what happens when you give fecal transplant to a patient with recurrent C. difficile? And again, this is just to show you the power of looking at community structures over time and the changes. So again, we're going to use this as an example for 16S sequencing using next-generation sequencing platforms, okay? So this was a collaboration that was started by a number of clinicians who had been doing fecal transplant for recurrent C. difficile for quite a while. And they had called me and they'd asked, well, how would you do this? How would you collect specimens? What would you do? I didn't hear from them for a few years, but when that paper came out, they said, hey, we have a bunch of samples. Can you sequence them? And we sequenced them quite quickly. And as was asked in the question, we generated this sequence in about a week. We isolated the DNA, did the amplification and sequencing, but then we had a data set sitting there. And luckily, that was the day that Anna showed up in my lab, and I was actually at a meeting where Johan Bakken was at the FDA and the NIH, where they're talking about regulating fecal transplant. And he says, hey, did you get the specimens? Did you generate data? I said, yeah, I generated data. Haven't looked at the data yet. This is 8 a.m. Called the lab. I said, Anna, welcome to the lab. By the way, could you go to the lab? There's a data set there. She'd already been trained during her PhD on how to do the analysis. So by noon, I was able to flip open my laptop, and I showed Johan, I said, hey, here's a preliminary analysis of the data. He goes, that's quick. How do you do that? I says, I didn't. I have good people working for me now. So let's go over these data, because I'll tell you about how we analyze these data. So these are the patients, okay? If you look at patients, the FMT marks when they got the FMT. They had multiple rounds of antibiotics. They had multiple positive tests for C. difficile. Where'd the pointer? Oh, here it is. They had multiple positive tests for, it's kind of dim. Anyways, you can see the multiple pluses for the positive tests there. And we got samples prior to getting the transplant and after getting the transplant. And importantly, in most cases, we also go to the donor specimens. If you notice, there are a few of those gray triangles missing from the early time points because the people would always throw it out. They never thought to save it, but eventually we said, no, it's very important to save the donor sample. So we isolated DNA, we did the amplification and sequencing of the 16S. But what do you do? Who's ever read a paper where they've done 16S microbiome analysis? You see all these terms, borrowed from ecology, Morse-Horn diverse index, operational taxonomic units, what's the inverse Simpson diversity? And you kind of go, what's going on? Let me take you through some of these data, because it's relatively easy to understand. So this is the figure that a form of this figure that I first showed to Johan. Well, one of the things you can do is you can look at database. We talked about databases for genomes. There's databases for 16S sequences. Basically, what we did is you can match it and kind of try to assign a name to that short snippet, 250 base pairs of 16S, and assign a name. And what you need to know is that sometimes the names are kind of funny. In yellow is Clostridium 11. That's actually how some of the classification schemes refer to C. diff. So they don't always give you the right name. They give you a taxonomically correct name, but not maybe the one that you're familiar with. But the important thing, they're grouped by patient. And if you look and not even care about what the different bacteria are, you can see that the sample pre, in most cases, is different from the sample post. And what it is more resembling is the donor. You can see that for patient 1, you can see it for patient 15, 14, just kind of look on down. So this is a way to say, hey, look, we did transfer the microbiota, and this is 2 weeks after they got the fecal transplant. There are other ways to look at it. You can, again, assign them to different families, different genera. And now, in this case, we clustered them based on community type. Okay, we just allowed the data for speak to themselves and said, okay, which of the different communities are the most like each other? And you see two major groupings. This kind of smaller group on the left in this node, and then this larger group over here. And when you break the code, in blue are all the donor specimens. They're in this more complicated side. Without even knowing what the species are, you see that it's more diverse. There's many more types of bacteria. This side has lower diversity. It's dominated by actually E. coli and related organisms. And all the patients, most of the patients prior to transplant are over here. And after the transplant, they move over towards this higher diversity side. But you can't use it as a predictor. There are 2 patients that didn't respond. And when I first saw this, I asked Anna, hey, these 2 that after their transplant were still in lower diversity, were those the 2 that didn't respond? And she said no. So that's an important lesson. You hear that maybe you can predict a lot of things on the individual patient from the microbiome. That's probably not true. There's some things you can see by looking at populations, but it may not apply to the individual. So I mentioned diversity. In fact, when I first got involved with this, I was at Michigan State University, and if you look, I was in the National Food Safety and Toxicology Center. So that's where I started, actually. My career was actually in the area of food safety in Campylobacter and Helicobacters and things like that. But I got interested in Clostridium difficile because what I had, and we showed that there is lower diversity in patients with recurrent disease. And so the same thing was seen in this recent paper that we published. So prior to, and it doesn't matter how you measure diversity. You don't have to know the details. You can look them up if you want them. But before the transplant, they're lower diversity. The normal donors had higher diversity as a group, but now we connected them. And in most cases, the patients after receiving the fecal transplant had higher diversity. But diversity is just a measure of a community structure. There's nothing inherent functionally in diversity. And so how do you begin to look at function? One thing you notice is that the people are quite different. So one of the things that's sometimes useful is you need a good model, and we're fortunate in C. difficile that we do actually have a good model, that rodents can actually be given C. difficile, first by treating them with antibiotics to make them susceptible. So we've published a number of papers where we've looked at the structure of the microbiota as it relates to C. difficile infection. We've varied it. We looked to see what happens over time, the establishment of colonization when you're exposed to spores, the germination, the growth, etc. And we actually learned what are some of the interactions between the normal members of the microbiota and how they may interfere with C. difficile. But again, that's something that's more related to bacterial pathogenesis, not necessarily related to what we're doing here. So how do we get back to what we're discussing? Well, as I mentioned, what are the functions? What can this complex community be doing to limit a pathogen? How can that happen? Well, you can speculate a lot of things. Well, they can compete for food, or maybe they can metabolize things, or they may directly produce other antimicrobial products, or maybe they signal through the host. So how do you begin to discern that? Well, to do that, you can't just focus on 16S, because 16S is really anatomy. It's not physiology. It just tells you who's there, how are they assembled, what is the anatomy of the community there, how do we look at the physiology? Martin already gave us one way. The same way you can look at the transcription of a single organism, you could look at the transcription of a community by doing metatranscriptomics. You can do proteomics. But what I'd like to tell you about a little bit is how to look at the metabolic function of the community. And in this case, I'm referring to the microbiome, not just the bacteria, but the host. How the host and the microbes can collaborate to create a metabolic environment. So this was work that's done by Casey Terrio, who's now at the NC State University in the vet school there. But she was actually wanting to look at not just the structure, but the function. And we did metabolomics. Again, this is mass spec analysis of the whole gamish that's found in the intestine, and trying to look both in targeted and untargeted manners at what are the small molecules that are present there. And these are small molecules that may be generated by the bacteria, or by the host, or both, as we talk about. And what Casey did is she'd actually taken one of our models of antibiotic-treated mice and found out that there are different states, depending whether or not you treat them with this given antibiotic, they become susceptible, but if you take them off the antibiotics and leave them on sterile food and water, they can actually become resistant again. And she wanted to know what's different. Well, the communities were quite different in all cases. Okay, the communities were quite different after you treat with antibiotics. So these are our normal mice that we never treated. These are the mice that we treated with antibiotics. Again, you kind of restrict to only a few different microorganisms. But interestingly, after 6 weeks off of antibiotics, you return to another stable state that is a different reassortment of what was there. So it's a different community structure, but it had the same function because they were both resistant to C. difficile. And she goes, well, that's kind of funny. You have different structures with the same function. Maybe if we look at the metabolites, we can find something that's interesting. And that's exactly what she found. All of the resistant states were similar in terms of metabolites. And we're just going to focus on lipids and bile acids because this brings us to the fact that the susceptible state was associated with an enrichment in torcolate. So what are bile acids? Bile acids are things that we make to help us digest lipids. All right? And without going into the details, there's many different forms of bile acids. We actually in our liver have them conjugated to amino acids. That's fine. And one of them actually is very good at making those C. difficile spores germinate. But microbes actually participate in the metabolism of bile acids. They'll remove the conjugations, they'll remove hydroxyl groups, and they'll even create certain types of bile acids that are actually toxic to vegetative C. difficile. So that you can imagine when you change the community of microbes by giving antibiotics, you change the bile acid flux through the gut, and this can actually interfere or facilitate the growth and outgrowth of C. difficile and thus resulting in disease. And this is what Casey's actually following up on quite a bit. And actually it's been looked at that by manipulating the bile acid pool, at least experimentally, you can actually restore colonization resistance to C. difficile. So how do we get back to food safety? So one of the things that's come up in the past 10 to 15 years is a number of people have looked and found C. difficile in foods, animals, and really want to know what's going on. So they've been found in swine, they've been found in calves, and the CDC conducted studies where you can actually find similar types of C. difficile both in humans and in food animals. So, okay, that's an association. What else is going on? Well, the CDC published, well, I'm sorry, I'll get to the CDC paper, but based on this out of England, they were actually doing whole genome sequencing, which we already heard about, to do source tracking of infection, okay? Same kinds of techniques that we heard about in the first two talks this morning. But the interesting take home here is that over half of the C. difficile cases, they could not trace to person transmission within the hospital. And so they were assuming it must be coming from outside. These are sort of community acquired. And perhaps it's on food, because you can find C. difficile on food. We can find the same types of C. diff on food that we find in people. And so that was being pushed a lot. But before we go too far, even though we can find some things with whole genome sequencing, in other cases we can find other things. Let's go back to cultivation, as we found out, trying to culture. So this is the CDC report where they actually used PulseNet and FoodNet to look at food for C. difficile. And they had almost 2,000 different retail meat products, and they found no C. difficile. So again, there's some debate here, there's some things that you can find, there's more work that needs to be done, but it's not just sequencing. You have to do good epidemiology, you have to do good cultivation, you have to understand what's going on, you have to do science. This is a tool that you can use to do science, okay? So, you know, I hope I was able to tell you about this idea of the microbiota, okay? It's very complex, right? It's part of a balanced ecosystem with us. It can go out of balance, we'll hear about what happens if it can go out of balance relating to diet. If we actually make it go out of balance by giving antibiotics, that can result in C. difficile. And then this is just, sequencing is one of the tools that you can use in sort of a systems manner to understand these complex interactions between us and our microbial partners. So all of this work was done in a lot of collaborative grants that we've had funded by the NIH to look at the microbiome, to look at C. difficile in a systems manner, and of course all the people in the lab who do the work. And as I said, I actually got my start, my first faculty appointment was in the setting of food safety, and it's great to be back, and thanks very much for inviting me to speak to you today. Martin.
Great talk. One quick question.
Yeah.
Numbers. When you have microbiota in the intestine is 10 to the 14, you deplete it with antibiotics, how far do the numbers go down, and how big is your transplant? How many organisms do you put in?
Right.
I've always had a challenge with figuring out the numbers on that one.
Yeah, so, you know, it's interesting. When we think about antibiotics, they don't sterilize. You know, we get 3-4 log kill. 3-4 log kill when you start out 10 to 14. That's still a lot that's left over there, right? So it's really changing, but that is a significant change, 1,000-fold, 10,000-fold. So functionally, you have to think of that as a big change. When we give the fecal transplant, we do it in an interesting way for C. difficile. We take feces, and if anyone wants to Google home fecal transplant, you'll find out how to do it at home if you want. And they'll have you take sterile saline, I don't know why it has to be sterile, put it in a Waring blender, and whip it up. Well, remember, 99% of what you have in your gut are anaerobes. Most of those are killed, so you're probably only giving a small amount, but it's enough because they are microbes, they grow. So the idea is that you put enough in there that can establish and grow up and hopefully restore the function.
What do you think about food production practices and how that might impact the diversity of communities that we're eating? Do you think that would have an impact on microbiomes?
That's actually been looked at, and as an infectious disease doc, one of the things we get concerned about are antibiotic residuals in food or additives that might be able to change. I don't know if Gary's going to speak to this, but there are certain additives and artificial sweeteners and other things that have been shown to change the microbiota in people and mice. I don't think we have enough data to know what the ultimate impacts of those are. But I think it's something that we need to keep in mind because there are potentially unintended consequences. For example, a colleague of mine at Baylor, Rob Britton, has found that trehalose, added to a lot of food, actually is very good at fostering the growth of C. difficile. And if you want to do association, the current epidemic of C. difficile correlates when trehalose has started becoming up to 50 to 100 grams of some people's diets. Again, that's just an association. We don't know. Something to think about. One more?
Interesting talk. I have a question. When you're trying to do fecal transplantation, aren't we increasing the risk of putting back multi-drug resistant bacteria through feces back into the person's lower intestines?
That is possible. We do screen for pathogens.
Not pathogens, I'm talking about multi-drug resistance because all bacteria in the feces have significant levels of integron sequences and multi-drug.
Yes and no. That depends. If you look at Gautam Dantas's work, there is the resistome, as they refer to it. It's there in some, not in all. We try to limit the amount of antibiotic exposure on the donors, but I agree with you. The ultimate would be to actually know what's missing, have these organisms fully genome sequenced, available, put together the appropriate cocktail to the person and give it back to, that's the idea of personalized medicine for the future. But again, this is not a medical talk, but that's the idea of what we'd like to get to ultimately, not just giving raw feces that's uncharacterized, so.
Thank you.
Sure.
Yeah, nice talk. Nice talk. Looking at the future also, in respect to the developing next generation probiotics, what are the various advances that this type of study can help in production of next generation probiotics?
I think that what we can use is that idea of, and probiotics is defined in an interesting way. You could consider feces to be a live microbial product when given in sufficient quantities can provide a health benefit. So, technically fecal transplant is a form of probiotic, but I think ultimately being able to understand what functions are necessary in certain diseases and how can we give those back in the form of one or maybe a small group of organisms, I think that's the kind of thing that we need to do. But we really have to understand the science behind the fact that something as crude as a fecal transplant can treat recurrent C. difficile.
Thank you.
Okay, thanks.